Okay, good afternoon and welcome. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research. And I'm pleased to welcome you here. I see many of my colleagues and Scripps members, but the Front Row Lecture Series is really designed to reach out our scientific mission into the broader community at large. And so welcome to those people from the community. How many people, is this your first time to Scripps? Okay, that's great. Welcome. And hold on to your seats. You're about to get a drink from the fire hose. So we're really excited. This is our third front row lecture, and our speaker is Ardem Patapoutian. But I thought that I would like to just say, since there's so many members from the community that may not be so familiar with Scripps and its mission, I'd say a few things about our institute. So this is a thumbnail sketch overview slide. I'll touch on the graduate program separately, but this is one of our two campuses. We have a sister campus in West Palm Beach, Jupiter, Florida. There are about 150 faculty here in La Jolla and about 50 faculty on the Jupiter campus. And the Florida campus is thriving. They are the third largest grant producing institute in the state of Florida. So they're outstripping the University of Florida, and we're really proud of our faculty there. It's a wonderful campus. If you ever have occasion to visit, it's a great place. We've established 5 areas of scientific emphasis that are represented by the academic departments that we have, and those departments are bicoastal. So there are mirror departments on each of the two campuses. We have chemistry, immunology, neuroscience, molecular medicine, and structural biology. And those are what we call the 5 pillars of Scripps, and our goal is to be in the top programs around the world in those 5 areas. We have a tremendous, prestigious faculty. We have Nobel laureates. We have 18 members of the National Academy of Sciences, and that's a really amazing number given the size of the faculty. So 10% of our faculty are now in the National Academy. What you're going to hear tonight is a lecture this afternoon is a lecture about basic research. But there's also a tremendous translational aspect to the programs at Scripps. And over the years, a number of high-profile drugs have come to market from things that started as basic research here at Scripps Research. So I think the elections are over, and now we can look forward to the return of drug commercials to television. And who has not seen a Humira commercial? So Humira was invented, the technology that developed Humira was invented in Richard Lerner's lab. Richard was our president for 25 years. And this is a drug that has many indications for autoimmune type diseases. And it's definitely in the billion-dollar drug club. So the antibody technology was developed by Richard that eventually led to this Humira drug. Ray Stevens' lab developed this peg-valiase inhibitor. This is, in fact, a sort of cure for phenylketonuria, which is some people can't drink diet soda because it contains aspartame. But this is developed by Ray. And then actually Jeff Kelly's down in the audience. Jeff is the inventor. Jeff and I came here to Scripps at the same time. He invented a drug and I didn't. But this was work done in his lab. And I remember sitting on his students' committees looking at small molecules binding to transthyretin. And now it's been approved for amyloid neuropathy in Europe, and it's in phase 3 clinical trials now for transthyretin cardiomyopathy. And so this is a tremendous achievement. Very few scientists get the opportunity to see something they thought of at the bench put into people and actually curing diseases. And it took 20 years. And so it's thrilling to see Jeff's program coming to fruition at this time. And then the other major blockbuster drug that we have coming down the pipe is ozanimod. And it has two indications, one for ulcerative colitis, and it's also got an indication for multiple sclerosis. And we are hopeful that that drug will be approved by the FDA in the next 12 to 18 months. So in addition to the tremendous basic science that's going on in all of these departments, we've had a tremendous impact on human health. And really one of the crown jewels here at Scripps is the graduate program. So we have a program that's a PhD program in the chemical and biological sciences. We have about 250 students, 200 here in La Jolla and 50 on the Florida campus. It was established basically by smiting the ground with a staff and saying, we will have a PhD program just about 25 years ago. And in a very short order, the program rose to be ranked in the top 10 in chemistry and biology. And this is something we're extremely proud of. This is a peer ranking by deans and department chairs around the country that U.S. News and World Report does. And we've been consistently ranked for the last 20 years. And our students are going off to pharmaceutical companies and universities and to other scientific careers. Most recently, we got a tremendous donation to our endowment, and it's a founding endowment by the Skaggs family, and so you can see their name is now affixed to the Hazen building across the courtyard. And and we hope to build this endowment such that the graduate students are paid for the majority of their time here at Scripps. Okay, so welcome to Scripps. I just wanted to say a few words of context. Now let me introduce our speaker. So Ardem is one of our really eminent scientists. He is a neuroscientist. He studies sensory neurosciences. And this is something that all of us can appreciate. We're talking about the sense of touch, smell, how you perceive hot and cold or pain. And he is a remarkable speaker, and I'll let him tell you his own story. But he has this terrific felicity of expression that lets you really understand at a visceral level what's going on, but also keeping the science really at a high level. So Artem got his PhD at Caltech and did a postdoc at UCSF, and he joined our faculty in 2000. He is a member of the National Academy of Sciences, and he is also a member of the Howard Hughes Medical Institute. So for those of you who don't know, this is a tremendously prestigious organization where maybe the top 1 or 2% of faculty around the country are supported by the endowment for Howard Hughes. And we're very proud to have him as an HHMI member here at Scripps. So I'll cede the stage to Artem. And I should also add, he's a pretty good runner. He's a lot faster than he looks. So please join me in welcoming Artem.
Thank you, Jamie, for that very kind introduction. I didn't know I looked slow, but I am actually very slow these days since turning 50 last year. It's been a challenge. But thank you all for coming. So I want to start actually by thanking the people in my lab. So these are the people who do all the work that I will tell you about. And they're, as you can see, a fun-loving group. But they're also brilliant young scientists, and they're consistent of research technicians, members of the very highly rated graduate program that you just heard about, postdoctoral fellows, staff scientists, and an administrative assistant. And actually, after the talk, they will wear special badges and name tags. So please go up to them and talk to them. They love to talk about their science. In addition, as you will see, it's a very diverse group, and I want to emphasize this point. And actually, the next slide, my administrative assistant, Angela, put together. This is actually, a pin is put in at the birthplace of each of the trainees in my lab. And as you can see, they're all over the world. And girls are in green, and reds are boys, so gender-neutral colors. But just to make the point that it takes a global effort to do science, and I find it my privilege to come in and to work with these folks every day. So actually, Jamie also, we didn't coordinate, but he touched upon this as well. And I just wanted to throw out, before I start, a couple of terms that we use in biomedical research. So we do basic science, which is obviously aimed at understanding fundamental biological questions, such as how do cells communicate, how do you form a memory. But there's also applied science, which uses this basic science to try to find solutions to problems like treating diseases. And I think there are a lot of institutes across the nation and the world that are very good at one of these things. But in my humble opinion, Scripps is one of these places that's really exceptional in both of them. So today what I will do is I will tell you from my lab's perspective, what basic question we're interested in. And that is actually very simple, but an ignored part of biology. So most cells communicate with each other with chemicals. You might have heard of hormones being transmitted from one cell to another, or a neurotransmitter. But we know very little about how cells communicate physical stimuli, such as temperature and pressure. And hopefully I will show you that these basic questions have applied application to clinics, such as for pain, deafness, hypertension, etc. So of course we rely on our 5 senses to make actually sense of our environment. And many of these senses, we understand them quite well. At the molecular level, we know quite well how we smell, taste, and see. However, as I mentioned, how we sense touch through these mechanical and thermal stimuli is something that we don't understand the fundamental of it. But I'm here to tell you that your sense of touch is amazing. So human tactile discrimination extends into the nanoscale. So this is kind of mind-blowing, but you can sense an indentation about 1,000 times thinner than a human hair. Beyond this, we rely on affective touch for our psychological health. These are 2 kittens, Coco and Puff, that we just adopted a few months ago. Beyond this, there are all these senses that are related to touch, but probably none of you have ever heard of it. So proprioception is the sense of where your limbs are compared to your body. You have stretch sensors in your muscles, and from just how much they're stretched, you can tell where your arm is, and this is how you can close your eyes and touch your nose. The other things you can do with this sense is do complex tasks like play a musical instrument without looking at your hands, as beautifully illustrated here by Emmanuel Axe and Yo-Yo Ma. Again, related to touch is the sense of temperature, and as you all know, we are very sensitive to our environmental temperature. So much so that a few decades ago, car companies came up with this idea that you can have dual zone temperature control so that the driver and 2 feet apart the passenger can enjoy very different climates. Now, I don't have hard data on this, but from my personal questions to people, it seems like at homes, what temperature you set the thermostat is the #1 argument point for couples living together. So this is very important. So the flip side of this innocuous sensation is pain. And acute pain is essential for survival. This is sending us messages that there are damaging stimuli that we need to avoid. However, in some circumstances, even when the stimulus is gone, we still feel pain. And this is called chronic pain and is indeed a major unmet medical need. For example, patients who suffer from neuropathic pain, there's no good treatments for them right now. So all of these senses that I talked to you about, touch, temperature, pain, proprioception, all of them are sensed by this fascinating group of neurons called the dorsal root ganglia. So they're in the peripheral nervous system. By that we mean it's not in the brain, it's outside the brain. These neurons are just dorsal of your spinal cord. All those neurons are labeled in a zebrafish embryo. Each bulb contains about a few thousand neurons. And I'm going to play you a video that shows how these neurons, during development, extend and find their targets. It's like a tree extending its branches. it extends. These are large groups of axons, perhaps from 50 to 100 neurons. They're all going out to innervate the rest of the body, including skin, muscle, and internal organs. So that's the macro scale. This is a high magnification image of a DRG axon growing out. And one of the other fascinating things about these neurons is how long they are. So most cells, as you might or might not know, are tiny. They're a few microns in diameter. But these neurons have to do something very fascinating. So if you take Yao Ming, who's 7.5 feet tall, one of his DRGs in the lumbar area, for example, has to send an axon that goes all the way down. Axon is a small part of the neuron that you saw growing, to its toe, and at the same time it has to send a collateral all the way to the brain, making this the largest cell in his body, as much as 6.5 feet long. So you're slightly shorter than Yao, but you still should have a very impressively long DRG neuron. So what we've established till now is that we have these sensory neurons that extend processes to the skin, where they would gather information for mechanical force and temperature and send it to the brain. What we're really interested in is to know what are the receptors at the tips of these neurons that sense this temperature and force. So in the next 3 slides, I'm going to give you some very basic information on molecular biology. So bear with me if you're an expert in the field. But this should help you understand the rest of the talk. As you all know, our genetic code is encoded by DNA. And all of your cells pretty much have the exact same DNA, about 22,000 genes in them. However, your skin cell will do something very different than your liver cell. And for this to happen, you need to, quote-unquote, express or turn on different sets of genes. And when you do this, what we call transcription, which makes RNA, which then translates, which makes protein, and these are the specialized workforce of your cell. So according to what proteins you express, that's the phenotype of your cell. And these proteins, some of them might be familiar to you. They're enzymes like insulin or structural proteins such as keratin. In our case, these are the sensors that I want to talk to you about, and they're called ion channels. So what are ion channels? Your cell is surrounded by a lipid called plasma membrane, and this is very nice because it protects the cell and keeps what it needs to keep inside it. However, the cell also needs to communicate with the outside world, and it has sensors on this membrane to do just that. So ion channels are fascinating, very fast signaling molecules that are actually very simple. They have a hole in the middle, so they have a pore, and they do either of 2 things. They're either closed, which means they're not doing anything, or they're open. And when they're open, they have 10 million ions travel through it per second per 1 protein complex. So this flow is enough from multiple proteins to actually change the voltage of the plasma membrane, which then as an electric impulse travels through the neuron and then gets communicated from one neuron to the other. So the easiest way to think about ion channels is this electric switch that turns on a neuron. I also wanted to give you a scale of what we're talking about here. How big is an ion channel? So a grain of rice is about 500 times bigger than a cell. This is a typical cell, let's say. And now the ion channel would be about 100,000 times smaller than a cell. Can you guys see this tiny dot? That would be an ion channel. And it's not to scale. This is the smallest I could draw on PowerPoint. Okay, so now we've kind of reduced the question of you guys are familiar with this neuron and ion channels. What we're looking for is ion channels that would be specifically activated not by chemicals, but by mechanical force and temperature. And the identity of them is not known. We have 20,000 plus genes. Which one of these encode these receptors? I want to also tell you about why it's important to find these. Well, I find them very interesting. I think to know how we sense things like touch and proprioception are very fundamental questions. I already highlighted there obviously is clear relevance to pain. But this is one thing I want to also highlight, and I'll come back to this. And that's the beauty of doing basic science, is that sometimes you're looking for X and you find Y, and that Y is just as interesting as X. So research takes us into unexpected directions, and that's a very interesting part of doing science. So this is going to be my outline of what I will tell you about. Jamie said I came to Scripps in 2000. I've been here almost 19 years. And the first 10 years, we studied and found the first cold-activated ion channel, help us sense how we sense cold. And because this is older work, I'll go through it very quickly. and then I'll focus mainly on the novel mechanosensors that we've identified, Piezo 1 and 2, and I'll tell you their role in touch and pain, in sensing blood pressure, and this surprising story on malaria. So if you remember your high school or college physics, you would know this, but John Updike puts it very elegantly. Cold is an absence of heat, and yet it feels like a presence. Why is this? Well, because of tryptamate. We identified tryptamate in 2002. This was the first cold-activated ion channel found in the human body. And the reason that we know it's a cold-activated channel because of this fascinating technique called electrophysiology. So it's a patch that you attach to part of the cell. It could have one or more ion channels inside it, and you can electrically record the opening and closing of these ion channels. And I'm going to show you an actual reading from this. This is a tryptamate ion channel being recorded as you're cooling the sample from 30 degrees to 18 degrees. That's from 85 to 65 degrees Fahrenheit. And as you can see, initially you see these single openings, but eventually you see multiple ion channels being open here. So this is actually pretty amazing if you think about it. You are looking at a single ion channel, and I told you how small these things are, opening and closing. We're recording that small of a current. So this is in picoamps. Just for reference, 15 picoamps is this many zeros and then 15 amps. And again, for reference, a 100-watt light bulb is 1 amp. So very sensitive instruments are needed to do these kind of recordings. We knew immediately that we were dealing with a physiologically relevant cold sensor because we found that tryptamate was also activated by menthol, which is present in mint leaves. And this product is actually used in a variety of over-the-counter product to give you this minty cool freshness. And it includes things like chewing gums and Altoids and Icy Hot and Vicks, etc. Another of these cold channels that we identified is called tryptamate, and it's activated by colder temperatures. So we think of it as a noxious cold receptor. There was one interesting difference. The activators of tryptamate did not have this cool sensation to them, but they actually had a burning, spicy sensation. I'll give you one example. The ingredient in Listerine that has that burning sensation is because of wintergreen oil, which is a specific tryptamate activator. And so next time you're using this product and your mouth is burning and you're convinced that you're killing bacteria, let me tell you, all you're doing is activating your tryptamate channels coming from your trigeminal and innervating your tongue. So this stuff is supposed to be painful, folks. If we give them to animals, they avoid it at all costs. But humans, being interesting species, we've learned to love them. And for those who enjoy sushi, for example, again, wasabi, mustard oil, horseradish, all of these activate tryptamate, as well as the pungency of raw garlic. So based on these studies, actually, in collaboration with Pete Schultz, our president now, and Ben Cravat, also at Scripps, we figured out that the reason all of these chemicals activated because tryptamate is a general sensor of pain, and it actually senses reactive chemicals. And based on this and many other studies, now pharmaceutical companies are making tryptamate blockers in the hope of active clinical trials in various indications of pain. So that's all I'm going to say about temperature, and I'm going to switch to mechanosensation. And absolutely nothing was known about how we sense mechanical sensation in mammals and humans. And Bertrand Coste, a postdoc who came to my lab about 10 years ago with the intent to identify this ion channel. He was successful, and now he's a professor back in Marseille, France. And he took a very reductionist approach to find it. And what I mean by that is this is a cell in a culture dish, and he comes to a cell and pokes it just like that. And then he puts an electrode and records from it. And if this cell has a mechanically activated channel, you would see this in a graph like this, that the more you push it, the larger currents you see coming into the cell. So after screening cell lines that have strong currents like this, he found one called Neuro2A. And then he made candidates and knocked them down. So get rid of the expression one by one, try to see which one's responsible for this. It's easy for me to say it. It took me 30 seconds. It took him a year and a half to do this experiment. But he found an ion channel that is responsible for these mechanically activated currents. And the first one that he identified from these cells was called FAM38A, which stands for unknown family 38 member A. There's all these genes in our genome that no one has studied before, so they give them this really silly names. And one of the cool things about finding a function for a new gene is you get to rename it. And Bertrand named this Piezo1 from the Greek piezi, meaning pressure. So there's two piezos because they look similar, piezo 1 and 2. I'm not going to talk about it, but in addition to in humans and other mammals, piezos are present in all animals, plants, even pathogenic protozoa. One of the characteristics of piezo is they're very large proteins compared to others. It has 38 transmembrane domains, which means that it has regions that go up and down this fatty plasma membrane that I talked to you about, and that is more than any other protein in your genome. So one of the big breakthroughs we had early this year, actually, is instead of drawing this cartoon, is find the real structure of piezo ion channels. And this is work done by Kei Saotome in the lab. And this is one of the first 4 brilliant postdocs in my lab that I'll tell you a little bit, vignettes of what they're doing currently. And Kei is a joint postdoc with my colleague, Andrew Ward, that we collaborate very closely with. And he used this method called cryo-EM microscopy to find the actual structure of piezo1. And this is really a transformative technology that right here at Scripps played a fundamental role in establishing, and we're right now at the forefront in the whole world of using this technology. And what it entails is taking purified piezo protein, putting it on a grid, freezing it, putting it in this massive microscope, and getting hundreds of thousands of images of piezo protein. So each of these, encircled by green, is a very low resolution, not very interesting in itself, image of the protein. But through various software uses, we can do particle image averaging. From this, we can build a 3D map. And finally, we can build a molecular model where we know exactly where each amino acid is compared to each other and the space around it. And amino acids, of course, are the building blocks of a protein. So this is not the high-resolution structure of piezo. This is Kei's arms. And just to tell you the architecture of what this protein looks like. It has 3 subunits. It means 3 protein segments come in to form one complex. At one end, the protein comes together to form the whole, which I told you is important for ion channels. And the rest are kind of spread out in the plasma membrane, we think sensing plasma membrane tension. So this is the actual real structure that Kei solved. It's a thing of beauty. It's rotating so you can see it. Each subunit is a different color. And one of the things you will immediately notice that it's kind of puckered upwards. It's kind of bent in a way that's not very normal for proteins. And we think this is actually one of the ways where piezo is primed to be activated by mechanical force of membrane tension. So that any flattening or stretch of the membrane would pull on these ends. And the protein also has these interesting beams intracellularly that connect the outside to the pore. So by simply pulling on it, we think it just opens the pore and ions flow in. This is one of the few great examples, I think, that structure immediately suggests function, and we're very excited to follow up on this. But the rest of the talk, I want to focus on function, not structure. And as I told you, whether a gene expresses a protein gives us a hint already that it's using it. So doing this, we noticed that piezo2 is actually expressed in our favorite neurons that sense touch and proprioception. So how do we ask if it's required for function? What's commonly done is that you find a way to get rid of the piezo protein and see what happens. So again, I want to remind you of proprioception because it's going to be very relevant for the phenotypes that we observe. Proprioception, again, is sensing muscle stretch. And I already told you this is how you can close your eyes and touch your nose. You might not be able to do this, but you could actually close your eyes and walk straight pretty well. And again, all of these are dependent on these sensory neurons that innervate the muscle. So when we got rid of piezo in animal models, like in mice, and I won't show you the behavioral experiments, but they completely lost touch sensation and they completely lost proprioception. What I will show you is at the cellular level, the phenotype that we would expect if this was indeed the case. Our collaborator, Katie Wilkinson, can go into the muscles and stretch them. And while stretching, she can record from the neurons that are firing. Each line is the neuron firing. As you can see in a wild-type normal animal, when you stretch the muscle, you see massive increase of action potentials. But from a Piezo2 deficient animal, most of the cases we see absolutely nothing. Once in a while, we see something tiny left. So this is, of course, fantastic. One of our goals is to identify the touch and proprioceptive sensors. But these are, of course, all animal studies, and we were very excited when a group at NIH, led by Karsten Bonnemann and Alex Chesler, who have become our close collaborators, identified humans that are deficient in Piezo2. And just like in the mouse models, these humans cannot sense touch, and they don't have proprioception. And this video is going to show you what that lack of proprioception does. This patient is blindfolded, and she's just simply asked to walk in a straight line. And she's wearing a harness so that she doesn't fall down. And as you will notice, she has a very, very difficult time walking without visual cues. So how does this research help patients? One of the interesting facts is that many patients who have gait or walking trouble and go into a doctor's office, they usually get misdiagnosed because most of the time they'll say they either have muscle problems or motor neuron problems. Sensory part is often ignored. And these patients actually did not know that they had sensory deficits, amazingly, till their genes were sequenced and they realized that they have a deficiency in the touch sensor. So just finding this gene, what causes their deficiency, is actually already helping them because you can do very specific treatments once you have this knowledge. The main one is kind of obvious. You have to use your visual cues to compensate for the lack of touch sensation. We're also hoping that the models that we've built that are similar to the human conditions will help develop future treatments for these patients. So brilliant postdoc number 2, Shweta Murthy, was interested in, well, does Piezo2 play a role in pain? We already knew that it does not play a major role in acute pain. The humans say they sense pain. Tests in the mouse show that there's small deficits, but overall they're normal. But we're very interested in a more clinically relevant pain called mechanical allodynia. And this is defined as a painful sensation caused by innocuous stimuli like touch becoming painful. So this is something that, as I might have mentioned, that neuropathic patients suffer from. You might experience this from a simple sunburn. After sunburn, just a touch or even wearing a shirt becomes painful, and that's what allodynia is. In the laboratory, we can easily induce this by adding capsaicin, which is the active ingredient in chili pepper, which causes inflammation and causes this mechanical allodynia. So Shweta, with Megan's help in the lab, did this experiment in mice that don't have Piezo2. So they rub a little bit of capsaicin on their paws, and then they look at the mechanical threshold. At what force by these thin filaments do the animals tell us that, oh, this is not very pleasant? If you look at the wild type, which is in gray, and this is just the normalized mechanical response of these animals, only after 5 minutes of applying capsaicin, you can see that their threshold has dropped significantly, and they find it much more painful just to touch. But look at the Piezo knockout mice. They have completely normal, they don't seem to care at all that you have applied capsaicin. What's again exciting is that parallel to our studies, our collaborators, Alex Chesler at NIH, has actually been able to do the same experiment on the human subjects who have graciously agreed to do this. And what he's doing is applying capsaicin on one part of the arm and a control vehicle on the other. They don't see which side of the arm is what's applied for it. They wait for 30 minutes and do touch test and simply say which one hurts more, A or B. They have a very nice control that they know everything is working. By using laser Doppler imaging, they know that where they apply capsaicin, there is inflammation, redness, because this is immune response, so it should happen whether they have Piezo2 or not. But here's the fascinating results. As controls, 100% of the time know exactly where the capsaicin application was. The Piezo2 deficient people could not tell. They couldn't tell the difference. When forced to choose one, they choose randomly. That's why if you average them, it comes about 50%. So this has very profound clinical implications. As I told you, neuropathic patients suffer from mechanical allodynia, and this really shows that mechanistically blocking Piezo2 should be an interesting way to ameliorate this phenotype. But also very interesting is the fact that most pharmaceutical companies have actually started getting away from making pain medications, with the reason that many of the studies that basic scientists do in animal models haven't translated into the humans in the clinic. But this situation gives us a great advantage because we now already know that this is conserved between human and mice. So till now, I told you about the role of Piezo 2 in touch, proprioception, and this specific type of pain called allodynia. But the last few years, we've also realized that we have lots of internal organs also experience mechanical force. And these same sensory neurons that innervate and give us the sense of touch and proprioception also innervate these internal organs. So for example, 2 years ago, we had a collaboration with Steve Lieberlitz at Harvard, and we showed that Piezo 2 is required to control breathing. And without it, Herring-Brewer reflex, which is a reflex that if the lung is inflated too much, you stop breathing, is gone. The next, I will tell you a very interesting story about how piezos also regulate baroreception, which is a neuronal way of sensing and controlling blood pressure. In the future, we're also interested in extending to studies to bladder, which is an obvious mechanosensory function, but also stomach stretch. So most of nutritional sensing these days is focused on chemical sensing through the stomach. How much proteins, amino acids, and fat are you taking in? But if you're like me, if you eat too much, the first thing you feel is that you're full. And amazingly, how much stomach stretch plays a role in how much we eat is something that's not been studied very well at all. And we think the reason is that the molecules responsible for them were not found. So the addition of piezos gives us the opportunity to ask these fundamental questions. So the third postdoc that I will highlight his work is Weizheng Zhang. And I hope no one from EHNS is in here because I'm clearly illegally drinking coffee in the lab, which I shouldn't be. And he's very interested in the baroreflex. This is a homeostatic negative feedback loop that controls your blood pressure that, again, unless you went to medical school, you probably don't know about it. So baroreflex functions at a seconds and minute ranges to keep your blood pressure constant, and this is how it works. If your blood pressure increases suddenly, your aorta will stretch. You have mechanosensors of the types of neurons I've been telling you about that get activated. And once they get activated, the downstream consequence is activation of parasympathetic and sympathetic nervous system, which would decrease the heart rate and decrease the blood pressure. So without this baroreception, for example, if you're sitting down and stand up all of a sudden, you'll have a drop in blood pressure, and without baroreceptor reflex, you will feel dizzy and you might even faint. So you use this all the time, but you might not know about it. And the long-term consequences of baroreceptor function, lack of baroreceptor function, is many cardiovascular events such as arrhythmias, resistant hypertension, etc. So Weizheng looked at baroreceptor function in many different ways. I'm just showing you one of them. He can, in an anesthetized mice, he can inject phenylephrine, which is a vasoconstrictor. So this would make the blood vessels constrict and blood pressure go up. This is shown mean arterial pressure and systolic pressure. And as you can see, this is the consequence of baroreflex. The heart rate immediately dips. One of the things we noticed is that these neurons, unlike the touch neurons that only express Piezo 2, have both Piezo 1 and 2. And then in this example, if you get rid of both Piezo 1 and 2, the blood pressure still goes up, but no change in heart rate. So this is proof that baroreflex is ablated in these neurons. This is one example, of course, and when Weizheng did this again and again in many different animals, he showed that Piezo 1 knockout by itself doesn't affect the baroreflex, 2 doesn't, but if you get rid of them, you see a massive deficit in the function of these neurons. I'm not going to show you, but these mice also have very high blood pressure variability, which is a hallmark of lack of baroreflex, and are hypertensive. And we think that these new basic information could potentially help future experiments to see if piezo-expressing neurons, modulating them, can be useful in the clinic. So as you know, when we find a new finding, we publish them in scientific journals. And this is a very recent study, actually. We published it just 2 weeks ago in Science. But there's really a new way to communicate science these days, and it's via Twitter. And I'm highlighting Eric Topol, who's the god of Twitter in science in a way. He has like 150,000 followers. And he tweeted that the baroreflex has been known since 1852, even some description back to 100 AD, but we didn't know the mechanism until today. Hello, piezo ion channels. And he followed this up by a typical day at Scripps when one of my colleagues solves a 170-year-old mystery. So if you haven't figured out why I'm showing this slide, let me just be blunt. It's a shameless way to show you my Twitter handle. Okay. So till now we talked mainly about all the mechanosensory roles of these somatosensory neurons that I told you responsible for pain, baroreflex failure, touch, and proprioception. But we've really gotten excited because we believe that piezos and other mechanically activated ion channels that we're identifying is a great way to understand lots of different biological processes and diseases that depend on mechanotransduction. So many cells experience, sense, and respond to mechanical stimuli. This includes inner ear for hearing, blood vessels. In addition to the neuronal sensing, blood vessels themselves sense pressure and respond to it accordingly. Bone cells experience mechanical force. Gravity is one aspect of this. If you might have heard that astronauts who go into outer space lose bone density. Red blood cells experience massive amounts of mechanical force, etc. So I won't have time to tell you other systems that we've studied today too much, but let me just mention that piezo 1 is expressed in blood vessels, and we've recently found that if you don't have piezo 1 in a collaboration with a clinical setting, that you get lymphedema. So your lymphatic system usually drains all this lymph away from your extremities, and in this case, as you can see in these patients who don't have piezo 1, they have swollen abdomen or feet, as in this case being shown. But the thing I do want to talk about, and this is what I initially mentioned of how research can take us in directions that we didn't anticipate, is role of piezo 1 in red blood cells. And this is the work of Shang Ma in the lab, who has a postdoctoral fellowship from the Gates Foundation through Caliber, the drug discovery wing of Scripps. So Shang has been studying hereditary xerocytosis, and this is a disorder of red blood cells, and they have dehydrated red blood cells. You can see the wild-type cells and the arrows pointing at some dehydrated red blood cells in these patients. And it's not a very severe disease. They experience anemia, but very mild and enlarged spleen. But many times it's asymptomatic. So there are some people who had it who don't even know that they have it till they get genotyped, for example. And it was thought to be extremely rare. There are only 100 cases worldwide, and all of them, though, and this tells you why Shang is interested in studying it, have mutations in piezo 1. However, the mutations are very different from what I told you before. The piezo 1 and 2 mutations I've been telling you about all are loss of function. They're not functional. In this case, they have point mutation, which makes them a little bit overactive in the sense that given a certain amount of stimulus, they signal more. So we got very interested in this from another perspective. You might have heard of sickle cell anemia, and we know that genetic conditions that are associated with resistance to malaria often have morphological deficits in red blood cells. So Shang asked the question, is piezo 1 gain-of-function mutation prevalent in, for example, African population where malaria is endemic? He found one, but the surprising thing was how abundant it was, which was really shocking. So this is showing this new allele that we found. It's called E756 delete. It's lacking one amino acid from the 2,500 amino acids that piezo 1 is composed of. And this chart shows the prevalence in different populations. The wild type is in this lighter color. E756 delete in this purple dark color. And it is a whopping 1 in 3 of every person of African ancestry has one copy of this, including African Americans. So this really surprised us. Partly because how is it possible that so many people would have slightly dehydrated red blood cells and not know about it? So we actually have gone in and checked directly. Sean got red blood cells from African American donors. He got 22 donors, and just like the numbers would predict, 7 of them had this allele. And when he just simply looked at them, he saw that all the ones that carry this allele have some dehydrated red blood cells. I still don't understand how this could have been missed till today. But to test our hypothesis that this might be beneficial to malaria infection, we collaborated with Elizabeth Winsler next door at UCSD, and she helped us show that if you have this allele, you're actually less susceptible to infection by malaria, at least in a petri dish, in a test tube, in vitro. So this just shows that if you give the agent that caused malaria to red blood cells of non-carriers, you see this is the kind of normalized level of infection. And you can clearly see that if you do it from subjects that carry this allele, this is significantly decreased. So if you told me 3 years ago that I would be studying malaria, of course I would have laughed, but this is the beauty of basic science. And where are we taking this? Well, we're very excited to know if in the field this is something that's really relevant. So we're collaborating with Kitkowski Group at Oxford University, who has epidemiological data. So he has a big database of the DNA and phenotype of people who are infected with malaria. And we want to see if you have this allele, if you're less likely to get the disease. And I'm not going to show you, there are some very encouraging preliminary data coming from these studies. Of course, this suggests the possibility that Piezo-1 activators will be very useful to treating patients that might be infected with malaria. But one other area that we're really excited about, I don't know if you've heard of this, but there's now very large databases of genome and phenotype comparisons, so that if your DNA is known and you know everything that's good and not good with you, then we can go back and ask, how is this allele of Piezo-1 associated with any other human condition? So, for example, cardiovascular functions that I told you Piezo-1 is involved in. So I think in the next year or so, we'll have lots of large data sets to tell us what other things are wrong in people who have too much mechanotransduction. So the take-home messages today are that we have, in the last 18 years or so, have cloned these novel sensors that help us sense temperature and pressure. These are novel ion channels that initiate this whole sensation of touch, pain, sensing blood pressure. And I've told you stories where uncovering these proteins has told us both about expected, unexpected diseases associated with this idea of mechanotransduction. And the last 2 thoughts I want to leave you with is that, indeed, basic science research labs are nurseries for future medicine, so I hope you support it, and also that Scripps is an awesome place to do science. So I need just to thank people. I won't read all the names, but these are the awesome people who did all the work that I presented to you today. I need to also thank our administration, including Pete and Jamie, for creating this environment to do science at Scripps, and also all our collaborators. As we study many different aspects of biology, we couldn't do all of this without help. And as you can see of what an awesome place Scripps is, a lot of these people are from Scripps. So thank you for your attention, and if anyone has any questions, I'll be happy to answer them. So we haven't looked at that yet. That's more of a central control. It happens because of things in the brain that piezos are not expressed in. But that's the beauty of the genome phenotype association studies. Again, we expect lots of interesting but unexpected surprises when we look at these databases. So we will look at all kinds of diseases if any of these are associated with the genes and the processes that we study it. But we haven't looked at it specifically.
In a way that makes affects
That's interesting. We've thought about this. It might be, we haven't looked at this, but if you remember I talked to you about TRPA1 before I talked about piezos that sense reactive chemicals. It's actually been shown that many of these agents of chemotherapy activate TRPA1, and this might at least contribute to the pain or discomfort that they cause. And some of the ideas have been to try to block TRPA1 to stop some of the adverse effects of these chemotherapies. Whether proprioception is involved in this is something that I haven't looked into, but I'll think about it some more. It's a good question.
If you can model some of the molecules that are part of these receptor chemicals, can you regenerate them?
Can you regenerate them? So tissue engineering is a budding field. People are starting to look into this. We're not quite there. The type of modeling that we do that's very useful, we're just starting this, is once you have the structure of the protein, you can model chemicals that would dock, that would bind to it. And as I told you, in many of these instances, where this is again early basic science, that if we had a chemical that blocks or activates piezo, it would be very interesting. And normally this is done by screening millions of compounds blindly and finding ones that would activate and block, and you would pursue it, and we're still doing that. But we also do modeling, and because you know the structure, that's a very useful part of it, is to see what molecules might bind to it just by modeling instead of screening, which is a much cheaper way of doing things. So we are pursuing this as well. Question?
Do you worry, considering that it affects the heart with the neuroreceptors, do you worry about manipulating levels of energy to bring it up, bring it down in humans?
Absolutely. That's something we think about all the time. And so, for example, when I suggested that blocking piezo2 might be very useful for mechanical allodynia for neuropathic pain, we don't think that this would ever be an oral medication, because if you take that and it completely blocks it, you will not sense touch, you will not have proprioception, it won't be a good thing. But the good news about allodynia is lots of times it's local, and although it's more difficult to make a drug that does this, you can do local application potentially that would circumvent all these problems. So yeah, because it's doing too many things, probably local application is going to be more viable than taking it systemically.
Hi, I'm wondering, food with the different textures might give you different sensation in terms of taste. And do you see any integration between mechanosensation and other sensory systems? And are these molecules involved?
Yeah, we've thought about this a lot as well. So texture sensing, a mouthfeel, is a completely mechanical sense. And we're not doing this specifically, but a couple of other groups are actually looking into this very carefully to sense if all this texture sensing is through piezos and mechanosensation. And I think it's probably going to end up being the case. As you know, the different tastes of sour, umami, sweet, bitter, none of them explain, for example, how fat tastes so sumptuous in a way. And why is that? And people think actually it's the mechanical aspect of it that's not sensed by the tongue taste cells, but the trigeminal neurons that express these sensors, including piezos. So we don't think piezos are in the tongue, in the taste cells, but there's this whole chemical sensing from the trigeminal that all the capsaicin or the mustard oil or the wasabi, all of these are actually trigeminal. You think of it as taste, but it's a trigeminal chemical sense. And all TRPA1 as well as piezos are all expressed in this system.
Can you shed some light on how the lipid environment influence the ion channels to activate or inhibit the function?
We know a little bit about this. It's still active work, but it seems like lipid environment has a massive effect on activation of the ion channels. We can take these proteins away from the cell and just put it on a lipid bilayer, and they function as mechanosensors. And we and others have shown that the composition of the lipid by itself can modulate the function. So absolutely, it plays a huge role. The exact language that it's used, or what is it about the lipids that it's sensing, is it the length, is it how much saturated it is, are things that are still work in progress that we don't know the answer to. Yes, we've thought about that too. Actually, last year, we just published a paper that's a G-protein coupled receptor that's mechanosensitive. G-protein coupled receptors are this other group of sensors that are actually sensors for taste and smell and vision. So it's very interesting to find that class of molecules to be involved in mechanosensation. Obviously, ion channels are the fastest sensing, and their identity that they have to be ion channels was argued by an argument of kinetics, because it can happen much faster than any other system. But as we're talking about other mechanosensory systems that could be much smaller, I can even imagine a kinase to be mechanosensitive, for example. There's some literature on this, but not much. By the way, the acoustics in this room is so amazing. I can hear the questions without a microphone so clearly. Such as NAVs for pain. So there's these ion channels that are activated by voltage, and there was a study a decade or two ago now that humans actually who don't have this channel don't feel pain at all, complete lack of pain, which is not a great thing. Many of these people who have these mutations have ended up accidentally killing themselves by, for example, jumping from a third-story building because they thought they were invincible. And so those channels are actually downstream of these channels that I'm talking about. So they are voltage gated. Remember how I said that the activation of these ion channels changes the membrane potential? So those ion channels, the NAVs, that come downstream for it. Once the voltage is changed, they're the propagation channel that carries the signal from the inception to actually action potentials. So as far as we know, they're not mechanosensitive themselves.
Okay. Oh, wait, one more. Yep.
Artem, the slide that you had up that showed malaria, for example, also had osteoporosis. And I wondered if your lab or labs you connect with are really focusing or are focusing on helping people that might have osteoporosis or are already working.
Absolutely. We have both collaborators, and our own lab now is turning off piezos in bone only and trying to see what the consequences are if you have too much mechanotransduction or not enough. Ask me again in 6 months.
Okay, let's maybe just. Yeah. So I have two things to say before we go, but first let's thank Artem for a brilliant lecture. So just quickly, two thoughts. I'm reluctant to cut the discussion short, but two thoughts. So first of all, I just want you to know that Artem is a star, but we have a pretty deep bench here at Scripps. And we encourage you to come and see the other front row lecture series that we develop throughout the rest of the year. And then the second thing I'd like to point out is that there was a number of examples of true collaboration between Artem's lab and other faculty at Scripps. And this is very much one of the hallmarks of the work that we do here. People look to each other to support each other and collaborate to do things that no one could do on their own. So we have a tremendous ecosystem here. So thank you for coming to our ecosystem and getting a glimpse of what goes on here. And we want you to continue the discussion at the reception out there on the plaza. So thank you all for coming.